My name is Michael Battelle, and I'm the president and founder of the Fatty Liver Alliance. We raise awareness about the risks, causes, and complications of fatty liver disease and help those already diagnosed with NAFLD and NASH by advocating for access to approved treatments and care. Today's very special guest is Rachel Zayas, the CEO of Aged Diagnostics. We met at the ASLD Nomenclature Roundtable in Washington, and I asked Rachel if we could have this chat today to learn more about her work. Rachel, what is Aged Diagnostics and why did you decide to start your organization?
Hello, thank you. Thank you so much for the opportunity to speak today. Excited to be here. Aged is a diagnostics company developing non-invasive diagnostic tools for NASH and liver fibrosis. We are a team of geneticists and hepatologists who started our pursuits in 2018, and we're just getting through some clinical studies to really prove out the concepts of our core technology. A little bit of history about me and history that ties into this company is I worked at MIT in cancer research for a number of years. And when I was there, I was about to start a PhD program when I realized that the tools we were using were not getting well translated into the clinic. So I was on the academia track and I had never taken a business class. And I said, there has to be a better way. There has to be a more effective way to translate these tools into the clinic so then patients can benefit. So I took a chance and I dropped out of the PhD program to go for an MBA. And it was the best risk of my life because patients can't wait and the market couldn't wait. And I knew if we sat back and waited for 7 years, this opportunity would pass us by. So I took a team of colleagues and we started this company, and it's been quite rewarding. And so here we are today. We're developing a couple blood tests for NASH and fibrosis, and I'll pause there.
No, that's okay. So on that note, your claim on your site, your dream, your hope is that you're going to be developing, or you're already in the middle of developing, one of the most accurate blood tests that's going to be available for NASH. So maybe just a little, can we have a sneak peek on your thoughts of why that is?
Absolutely. Excellent question. So I have a genetics background, and we saw emerging trends about 10 years ago with the epigenome. So if you look at a lot of blood tests, they focus on genes, proteins, transcriptional expression, but these are not as reliable, sensitive, or specific as the epigenome. When the epigenome reorganizes, these patterns are more specific. And I just want to break down what that means. So we all know the word genome, a string of DNA. Epi is a Latin term that means above. So the string of DNA doesn't change, but what's changing is the methylation status or the methylation pattern above the genome. So the DNA remains intact, but the methylation or the expression level of that DNA changes over time. And this has been known to be more specific for disease, health, wellness, longevity, inflammation patterns. And there's been a lot of buzz and a lot of research in oncology, but it's now starting to get translated into other fields. So 10 years ago, this was really cost ineffective, but there's this theory called Moore's Law, where the cost of sequencing has gone down twofold every year for the last 25 years, just because of the computational power that we have, that we've been developing as just a field. So we saw these trends emerging and we said, well, we want to determine which epigenome patterns are most sensitive, specific, and reliable. So we saw these trends emerging 5 years ago, and we wanted to be the first to market and develop a team that can utilize these resources and translate them well.
So, and if I missed it, I'm sorry, but I want to ask it just to be clear. So would this test be something that is done early on before somebody may have disease as a predictor of disease, or is it over time genetically we change and that's what you're detecting?
This would be a tool that can differentiate various stages of NAFLD, so being able to differentiate simple steatosis from the early stages of NASH, and then a second tool that can stage liver fibrosis. So or like, well, our goal is when we launch, it's to work with gastroenterologists and hepatologists because they're keenly aware of the problems. our long-term strategy is to get this into the hands of primary care physicians, pediatricians, internists, to be able to routinely monitor their patients at an early stage before there are any complications and before there's a need to even get into a specialty clinic.
Okay. So you'd have a baseline when a patient comes in to see you, and you would store that. And then what you do, serial tests, would it be like an annual test, you think? Is that what you foresee?
So we would hope to have a baseline, but in most instances, it's
Okay.
the standard is off of the general population, and our hypothesis is that it might be most effective to screen patients every 2 to 3 years.
Okay. All right. Okay. I asked the earlier question because we're coming up to this pediatric liver disease month, and I was wondering, hey, this would be so awesome if you could test somebody when they're like a baby and see if they have a chance, like a higher predisposition for developing NASH in time. But I understand. So this is in response to changes in your body.
Yes,
Okay.
yes, precisely.
Okay. So you believe in early assessment as a that's one of the drivers of why you're doing this, right?
Absolutely. I think that if we can get this knowledge into the hands of physicians and patients to make lifestyle modifications and have very intentional screening programs, this can make all the difference. Because if you think about it, just by the sheer number, not everyone can get into a hepatology clinic. If most patients, if even just 5% of patients are going on to develop major adverse liver outcomes, they're not going to be able to, just by the sheer number of patients versus providers. We need to intervene earlier on in the pipeline, and change the trajectory into the primary care clinic as we can.
I mean, there's no question non-invasive testing is the future. As you say on your website too, biopsies have complications and challenges. And so I do believe that in the real world, as you and I talked about offline, that the future is really going to be in these types of diagnostic blood tests, probably, as well as ultrasound-based scanning technologies. Do you see that the Aged Diagnostics test would be used in conjunction with a visual scan test like that together? Yeah.
Absolutely. I don't think there's going to be one specific tool that can replace everything. I think it's a sequential testing strategy. So I can see this reinforcing MRIs, MRE, CAP, et cetera, and making an informed decision from a synthesized assessment.
Okay. What would be your takeaway message that we want to share with people before we say goodbye today?
That there is a distinct difference between the genome and the epigenome. So we're not looking at inheritance patterns. We're looking at changes that occur in your individual lifetime. So I'm a big advocate for just sharing this knowledge in genetics, because the more we talk about it and the more we break down these silos, the easier it will be to translate these tools into the clinic. So that's my one takeaway.
That's great.
Message.
Super exciting. Looking forward to progress updates, and we'll have to have you on again when we get closer. Anybody, can you hint at all? What are we looking at with timelines, by the way? How far?
So we're actually launching a large trial this year, 400 patients. It's a large research study, and then we expect to launch by 2025, which somehow is around the corner.
It's going to come really fast. We're going to mark our calendars. That's great. Thank you so much for being with us today and sharing this information for patients.
Definitely. Thank you for the opportunity to be here.